Hi, everyone. I'm Shane McCarthy. I'm at the University of Cambridge and the Sanger Institute. I work mainly on the DTOL project on the assembly side of things. Welcome to the Sequencing Technologies and Techniques section. We have 4 talks today. We'll have the talks and a brief Q&A after each one. And then so for the questions, they should go in the chat at the bottom and upvote, and actually try and add your questions during the talk if you can, so that they appear when we switch over. And that's it. So first up is Jeremy Schmutz from Hudson Alpha and JGI, talking about ultimate plant reference genomes.
My name is Jeremy Schmutz. I'm from the Hudson Alpha Institute for Biotechnology in Huntsville, Alabama, where my group mostly works on plant genetics and genomics. I'm also part of LBNL at Florence Berkeley National Lab, the DOE Joint Genome Institute, which is a user facility for the United States Department of Energy. It works in energy and the environment and genomics in those. So I'll be talking a little bit about data that comes from a few different experimental groups, including our one here at Hudson Alpha, which is run by Jane, and then also the group in California at Lawrence Berkeley, run by Yuko and Chris, that produce the data. And a lot of the projects I'm going to be referring to today are from the projects that have come through the plant assembly and annotation group here at Hudson Alpha and the JGI. So just a brief history of plant de novo genome sequencing. We started out a long time ago sequencing very expensive genomes, about $10 million genomes for something like a sorghum genome in the Sanger days. Moved into 454 short read sequencing hybrids with Sanger, and then on to complete short read genomes with Illumina, and then back up into start to get longer genomes from PacBio RS2, and then longer genomes, higher quality genomes still from the Sequel and the Sequel 2s. And so we've gone in the course of just about 15 years from $10 million genomes down to $10,000 genomes, about 1,000 times reduction in the cost of genome sequencing. So these are my ratings for the years. I spent a lot of time sequencing genomes. These were definitely dark years from about 2008 to 2014 as we started to pick back up in genomes. So to see what this looks like in the context of something like a sorghum genome, this is our original Sanger whole genome shotgun genome here. In 2007, I threw on an all-pads Illumina build, so you can see what the SAD Illumina days look like for whole genomes of 50,000 contigs with a 0.02 megabase contig N50. And then this is our finished improved version where we finished gene space up to 1.3 megabase contigs. And then here is early PacBio. We were real excited in those days to get 400 KB contig N50s back out again. And then in recent years, 2018 here, the Sequel genomes where we're running something like 17.7 megabase contig N50 with about 411 genomes. So this has been great. We're starting to get really high quality plant genomes that are complete back out again from PacBio. But so I guess the question is kind of what's next? And what's next is PacBio CCS, or HiFi as they call it, where you're actually sequencing the same strand multiple times, then generating a consensus sequence. The important part here is that every HiFi read is actually a single consensus sequence generated from the same molecule. So there's no sharing, there's no all against all read comparisons for error correction. And we were part of the original Sequel 2 early access group where we worked on, among other things, developing experimental use of HiFi sequencing. And you can see kind of over time the increase in yield, but effectively at this point, we're about 15 to 25 gigabases per cell at 99.6% higher accuracy. So this has led us to a new improved CCS assembly paradigm where you do the upfront cost of the compute, which means that when you're doing comparisons, you can move into exact k-mers to increase the speed using some of the new assemblers that people have continued to improve. HiFi ASM, for example, for inbreds and diploids, and Canoe 2 for polyploids with Rakan for polishing. We've been able to take these genomes and move into Hi-C or genetic maps to really produce high quality genomes, followed by a little bit of polishing, consensus polishing from Illumina. So this has really enabled some amazing things to happen in the last year and a half. Here's an example of sorghum HiFi with our V5 sorghum, which is based on CCS, moving down to 89 contigs with the 50 meg contig N50 effectively. Nearly whole chromosome arms are coming out of these assemblies these days. So HiFi CCS gives us great advantages. One of them is to lets us assemble more difficult regions. Also lets us detangle some complex haplotypes and outbreds and polyploids. And it really greatly decreased our computation time, which in some cases can be 50 times less computation. So that means we can rapidly cycle through assemblies and really clean things up. This is kind of what it looks like if you're looking at gene space here, syntenic gene space. You can see we're still containing a lot of the same genes we had back in the Sanger WGS days in the CCS HiFi, but it's just cleaned up really nicely over time. We've actually added 5,910 genes since V1, and V5 also adds 2,057 private genes. And this is a representation from a program one of my folks is working on, John Lovell, called GeneSpace. And you'll see a few of these as we go through this presentation. So one of the reasons we're adding new genes is because we're adding a lot of IsoSeq data from Sequel II. Enables us to collect somewhere between 8 and 20 million full-length cDNAs from there. Shu and the annotation group has been working to integrate this and produce higher quality reference genomes annotations. So you can see here, we get about 7% more genes in a direct comparison for Brachipodium, one of our JGI flagships. 18% more transcripts. Most of these are coming from alt transcripts and alternatives validated by IsoSeq, and about 0.7% on our BLISCO score for this Brachipodium genome. So one of the other issues that we run into as we move outside of this inbred crop space is heterozygous genomes. This is an example talking about Pecan, a project we're working on to generate genomic reference and infrastructure for pecan breeding. So one of the issues that we have with outbreds is we typically end up with 2 copies of everything, and then we have to go through and sort these out meticulously to figure out how to get a single haplotype out. So our new solution to this is using CCS and assembling with HiFi ASM, which is a really nice ability to split those haplotypes out and give us big, long chunks to use. And so just to show you a quick example of what that looks like, we've gone from early Illumina days with 130,000 contigs down to long read pecans at about 500 contigs at a 4.4 megabase contig N50 to our latest version of Pawnee, which is a CCS, 34 contigs at 26.5 megabase. This one here is not our genome. This is Illumina PacBio hybrid. You can see that continuity is pretty low. So then moving on to a complex case, this is recent tetraploid, which is a domesticated cotton. And just to put on quite a bit of examples from the original days of whole genome shotguns and pooled backs genomes all the way down through, this is a de novo magic style genome, through our versions where we've worked on RS2 and long reads. That was early PacBio here at 0.78 or 780 kilobase N50 to 314 megabases, or 314 contigs at 40 megabases, which is our long read Sequel version. And then CCS, where we're looking at something like 70.8 megabases. And you can see that we've maintained mostly the same kind of genes over time. You can see a little bit of differences here where we've had some discussions about how paracentromeric regions are supposed to be organized in each one of these genomes over time. Notable here is that the NR version here has about 10,000 genes, which don't seem to match to the rest of the genes. And that's because of the high fracturing in that genome contig-wise that causes a great reduction in ability to predict genes. And then finally, new barley genomes are on the way. Here, working with Nils and Martin to try to generate high quality barley genomes. And I put this up here not as an example of showing that we're doing pretty good here. Nanopore and long read are both in the 10 megabase plus continuity, but rather moving on to CCS here at 25, but rather that there's a great reduction in remaining SNP and indel errors as you move through these technologies. And one of the advantages of CCS is it gives us about 7 times less errors in the consensus without polishing. And that means all those repetitive regions of the genomes are also higher quality than what we're getting out of PacBio long read or Oxford Nanopore. A lot of our data is available publicly at Phytosome. Phytosome-Next is the current version with 227 genomes from 123 plant species. In addition to the normal browser tools that are available, we also have added several different downstream analysis systems attached, including Centinane orthology, gene expression, now available for single cell RNA assays also, expanded gene family trees and profiles, and new clustered heat maps so that you can analyze the collection of data that we have available, make available publicly as part of the JGI. So how do we get to the ultimate plant genomes? We start with really good DNA. Here we source this from work with Dave Kundra at Arizona Genomics to produce high molecular weight DNA. We sequence it with CCS Hi-Fi on Sequel 2 and use new assemblers. Add Hi-C in assembly, curation, and polishing, and then use lots of RNA-seq and full-length cDNA for annotation, plus using advanced annotation pipelines. And that's how we get to the ultimate plant genomes. If you're interested in working on a Department of Energy relevant project, you can go to jgidoe.gov user programs, where we have several different programs that you can apply through and go through a review panel, and then you get to work with us on genomes and genomics related to energy and the environment. Just a few contact information, JGI user programs, which I just said. If you're interested in DOE relevant plants, feel free to shoot me an email. Biomolecule extraction, contact Dave. And then for non-DOE related sequencing projects, our Hudson Alpha contact is Jane Grimwood, hudsonalpha.org. And then for phytosome queries, there's the address there you can send it to. And so this is the work of a lot of different groups. I'm just highlighting some specifics about assembly and advances we've been able to make in plant genomics over the last few years. So there's a lot of people that are involved here in these projects that contribute. And then a significant amount of our funding comes from the U.S. Department of Energy, which funds us as a user program. Thanks, appreciate the time. Let me know if you've got any questions.
Thank you, Jeremy. Great talk. Let's open up for questions. Let's see. Nothing, yeah, so people put your questions in. One, let me start by asking, so with the polyploid and the CCS, do you have any sort of general rules for how close the haplotypes can be to be able to separate them? Have you found anything that way yet?
Yeah, for sure. It looks like the vast majority of haplotypes, wild haplotypes we've sequenced across things like trees, for example, are easily separatable. There's usually about 5% of the genome that really can't be separated, but mostly that's because it's made up of shared repetitive content between the haplotypes, not necessarily kind of genic space. For the most part, genic space separates really nicely. It's a little bit more difficult in vertebrates, in something like the human space, where there are definitely difficult parts of the genome that don't separate out, and that's because the cumulative errors in CCS, although there's not a lot of them, accumulate to the point where when the haplotypes get very close together, you can't tell the difference between a CCS error and a true variant between the two haplotypes. But for the most part in the plant world, almost everything is separable with CCS.
And here's a question, an anonymous one. We're seeing here that PacBio overall seems to be much better than Illumina in several aspects. Do you think Illumina will eventually die off and be replaced?
It's a question people ask a lot of me. I don't think so, personally. We run quite a bit of Illumina and quite a bit of PacBio sequencing. There is nothing like being able to generate 3 or 4 terabases of sequence data a day. Accurate data for things, and linking the difference between, on the resequencing side, de novo sequencing. I wouldn't do it any other way right now other than PacBio, CCS Hi-Fi. For sure, there's nothing else that we've worked with that is better.
Great, thanks, Jeremy. We're going to have to leave it there and move on to the next talk. Thank you.
No problem. Thank you.
So next up is Aida Verdes from the Natural History Museum in London, talking about visualization of toxin gene expression patterns in ribbonworm tissues.
Good afternoon, and thank you very much for having me here today. I am going to talk about a novel approach that we are implementing to sequence and visualize genes into tissue sections. This approach is called spatial transcriptomics, and I will show how we are using it in particular to identify putative novel toxins in ribbonworms, a lineage of venomous invertebrates that hasn't received much attention to date. So ribbonworms belong to the phylum Nemertea, and there are around 1,300 different species that are mostly marine benthic organisms, although there are some terrestrial and freshwater species as well. They are mainly characterized by an irreversible proboscis, which is this glandular structure that you can see here, and they use this structure to hunt and inject toxins into prey. The proboscis, when the ribbon worm is resting, is lying inside this coelomic cavity called the rhynchocele, which is also exclusive of the phylum. But Nemerteans don't have distinct multicellular venom glands. Instead, toxins are produced in secretory cells that line the epithelium of the body wall and are used for defense, and the epithelium of the proboscis, which I already mentioned, those are used for predation. So apart from this, we don't know much about ribbonworm venom. We don't know much about the diversity of toxins in this group. There have been just a few small molecules and some cytolytic and neurotoxic peptides identified, but only a few species have been investigated. So there's still much that we don't know. So we are using different approaches to look into the diversity and evolution of Nemertean toxins, including RNA sequencing-based differential gene expression analysis, proteotranscriptomics, or MALDI imaging. But one of the problems that we have is that we can actually milk these organisms to analyze the venom. So this makes it more challenging to identify putative toxins. And this is why we looked into spatial transcriptomics. Spatial transcriptomics is a technology that allows the visualization of gene expression or mRNA molecules on tissue sections. So the technology is performed on glass slides that contain capture probes, and each of those capture areas, each of those has 2,000 spots. Each of these spots has millions of spatially barcoded capture probes that bind mRNA molecules. And because they have this spatial barcode, we can then map each mRNA molecule back to its original location on the slide. So the process, very briefly, starts with frozen tissue that we section, and then we place onto a spatial transcriptomic slide. We stain the image and we stain the tissue and image it, and then we permeabilize it so that the mRNA falls into the spots and binds to the capture probes. We then perform cDNA synthesis on the slide, prepare libraries for Illumina sequencing, and we sequence them. Once we have these sequence libraries, we can analyze the data and investigate spatial gene expression patterns. So we optimized this technology to work with Nemerteans. We used it to investigate spatial gene expression patterns in the species Cerebratulus marginatus. And we analyzed transversal sections of the anterior part of 3 individuals. We stained the tissue sections and made sure we could identify the main histological features, especially those that we were more interested in, the proboscis over here and the epidermis, the ones that secretes the toxins. Once we finished the spatial transcriptomic protocol with these samples, we ended up with an average of 57 million reads, which corresponded to 3.5 million unique mRNA molecules that represented around 73,000 genes. Now, with this data, we can start to look into spatial expression patterns. So after processing the data, including some filtering and normalization, we cluster it according to gene expression patterns. In this plot, you can see that we identified 8 clusters, and there are 2, number 1 here and number 6 here, that are particularly different. But the most exciting part of spatial transcriptomics is that we can actually map these clusters on the tissue image, so we can correlate them with particular histological features. When we do that, we can see how the majority of the clusters mirror the major regions in the ribbonworm section. So for example, we have cluster 2 right here that corresponds to the stomach, or we have cluster 7 in bright pink that corresponds to the lateral nerve cords. And something we found exciting is that the 2 clusters that were quite different actually map to the two areas that we were interested in. Cluster 1 that corresponds to the epidermis, and cluster 6 that maps to the proboscis. And this is exciting because it might suggest that toxin genes that are not expressed in other places are driving this expression pattern. Because the data is clustered based on differential gene expression, we can actually look at the genes that are upregulated in each cluster. For example, we have here cluster 7 that corresponds to the lateral nerve cords, and we can see that most of the upregulated genes are related to the nervous system, which validates our approach as we are seeing genes being expressed where we would expect them. So if we look at other clusters that we are interested in, we have here cluster 1, the epidermis, where we can see that among the most expressed genes, we have some of the few known Nemertean toxins that have been characterized from toxic mucus. We have neurotoxin P4 and cytotoxin A3, and their expression seems to be restricted to the epidermis. We can also look at cluster 6, which represents the proboscis, and we see how there are lectins, metalloproteinases, and mucins that are upregulated, correlating well with the function of the proboscis of secreting toxic mucus for predation. We also found a basotoxin homologue, which is interesting because these type of hormone-like peptides are quite common in consnel venoms. But as you might know, if you work with non-model organisms, is that the majority of the sequencing data we obtain cannot be annotated. So in these clusters, there are also many other upregulated genes, but we don't know quite well what they are. So we started to investigate these novel upregulated genes in the proboscis cluster in particular, because they might represent novel predatory toxins. We found several transcripts like the one here that have a signal sequence and have cysteine patterns similar to other known toxins like cone snail toxins, and that are almost exclusively expressed in the proboscis. So these represent novel predatory toxin candidates that the worm might use for predation. We found other interesting transcripts like this one here that also has a signal sequence and belongs to the Huwen toxin family, which is a spider toxin family that functions as an ion channel modulator causing paralysis. Looking at the distribution of this toxin in the tissue, we see that it is highly expressed in the proboscis, but also in the stomach. So this indicates that this toxin might also be involved in the digestion process, and that ribbon worms might also use toxins to keep prey paralyzed once they've been swallowed. So these are just a few examples of what we can do with spatial transcriptomics technology and how we can demonstrate that it can be used to explore spatial gene expression patterns in ribbon worms and actually in other invertebrates as well. We have used this technology successfully to identify putative novel predatory toxins that are expressed exclusively in the proboscis of Cerebratulus marginatus. And we have seen how we can identify the expression of these toxins in other structures. And in summary, the spatial transcriptomics technology has a great potential to investigate venom in challenging organisms, but also in general to explore gene expression patterns. And before I finish, just wanted to thank our collaborators, funding bodies, and all the members of the Giacomello and the Riesgo Lab for their help and support. And thank you all very much for your attention. I will be happy to answer any questions. Thanks.
Thank you, Ida. That was a very interesting talk. We have a few questions here. Eric Jarvis asks, this makes me think that spatial transcriptome data would be great for genome annotation. Have you tried to or considered this?
Yeah, that's a very good question and very good comment. For these species in particular, and ribbon worms, we don't really have a genome yet, unfortunately. It's proven a bit difficult to assemble one. But yeah, sure, it's something that can give a lot of information to annotate certain genes. And the other people that use this technology, they use it with human tissue or like model organisms, mouse and things like that. And yes, it is helpful for annotating certain genes in the genome. Yes.
And Elaine asks, or she comments that that's a lot of unique genes. Do you have any idea how many genes are found in this worm?
Yeah, again, no no genome available yet. But the estimates are around 3 megabases. So there should be a lot of genes. But yeah, we don't know much about it, and most of them are cannot be annotated, unfortunately. But yeah, using some of these technologies, we can pinpoint a little bit more what functions might some of those genes have.
Great. Thank you very much, Ada.
Thank you.
In the interest of time, let's move on to the next talk. So next we have Xuan Cheng from the University of Sydney, talking about new long read based MHC typing in Tasmanian devils. Thank you.
Hello, everyone. My name is Xuan Cheng. I'm from the University of Sydney. Thanks for tuning into this talk. My talk today is about the use of PacBio sequencing for improving the efficiency and accuracy of MHC typing in the Tasmanian devil. The MHC is a gene cluster that's centrally involved in the vertebrate immune system. MHC class 1 and class 2 genes are the most polymorphic genes in humans and many other species. And in wildlife and conservation research, MHC diversity is widely studied as an important indicator of the immunological fitness of a population or species. Due to the rapid evolution of MHC genes, which often leads to lineage-specific gene expansions and diversification, the assessment of MHC diversity can be quite tricky in some species. And we have a perfect example for this, the Tasmanian devil. The devil is an endangered marsupial species in Australia. The species is threatened by a rare transmissible cancer called devil facial tumor disease. In order to better understand host immunity and evolution, as well as to inform conservation and management, it's quite important to be able to accurately genotype Tasmanian devils at their MHC genes, which is not as simple as it sounds. One of the challenges is that the MHC class 1 region of Tasmanian devil contains several structural variants. So what I'm showing here are the 3 MHC class 1 genes in the devil reference genome. So we found that due to deletion, one of the genes, this one, can be missing in certain MHC haplotypes. And it's not completely missing, but partially missing, which makes it even more tricky when you try to determine the number of functional alleles in the individual. And another even bigger problem is that the 3 genes share 98% sequence similarity. So it's basically impossible to design gene-specific primers. So if you want to genotype the genes by Sanger sequencing, you have to do cloning, which is very time-consuming and expensive. And also in this case, Illumina sequencing is pretty much helpless because the reads can't be confidently mapped to the genes that they came from, and it's pretty much impossible to accurately assemble the reads to their correct phase. So this has been a problem for us for quite a long time until recently, when we developed a new typing protocol based on PacBio amplicon sequencing. Basically, we designed a pair of universal primers that target all 3 MHC class 1 genes from intron 1 to intron 4. So the amplicons cover all the important functional domains. Of course, the primers are each tagged with a PacBio adapter to allow for barcodes to be attached during the second round of PCR. After that, up to 96 samples can be pulled together. Then there's just regular SmartBio library prep and sequencing. Using this protocol, we were able to genotype 384 samples over just a couple of months. So this protocol is pretty fast compared to what we used to do. Now, the data analysis can be a little bit tricky for some parts. First, for the CCS calling, usually what you do is to take all the subreads from the same SmartBio template and create a single consensus sequence. But because MHC alleles can sometimes be quite similar to each other, some alleles can form this type of heteroduplex SmartBio template. So to give you a real example, these are subreads from the same polymerase read. So each line represents a subread, and they're colored by strand. So quite obviously, the forward strand sequences and the reverse strand sequences represent two different alleles. So if you call consensus from all the subreads as usual, you can end up with a mosaic CCS read, which becomes extra noise in the data set. So the solution is quite simple. Instead of calling CCS with both strands, we did CCS calling for each strand separately. So basically we end up with 2 CCS reads per polymerase read. Another thing that we have to tackle is the allele calling or sequence variant calling. Although PacBio CCS reads can be quite accurate, they're still not error-free. And because we're trying to separate similar MHC alleles that can differ by as few as 1 base pair, we can't rely on clustering-based method to further polish the CCS read. PCR chimera is just another annoying common issue associated with amplicon analysis. And in our case, chimera is particularly prevalent in double MHC amplicons because double MHC genes have these extended regions with very high GC content, which are prone to form breakpoints for chimera formation. So we developed a tool to deal with these problems. So for example, what you're seeing are CCS reads from a single individual. Again, this is visualized in IGV to help explain our strategy. So what the program does is firstly to identify all the variable sites. Because we're dealing with CCS reads at this stage, it's not very hard to differentiate between real variations and sequencing errors like these ones. So in this example, you can see 6 variable sites. Then all the invariable sites will be masked and ignored, and only the polymorphic sites will be used for variant calling. So basically what we do is we take all these different combinations of nucleotides at the variable sites and to rank them based on their read count. The top 2 most abundant variants will be called real first. Then the program will go down the list and check each variant to see if it can be formed by any pair of variants before it. So over here, number 3 would be called a real allele, whereas number 4 would be called chimera because it can be formed by combining the first half of variant 1 and second half of variant 3. And same goes for the other 3 variants. This tool has been given a very nice nickname, Bellerophon, which is apparently the name of a Greek mythology hero who killed the chimera monster. So this slide summarizes our analysis pipeline. It starts with CCS calling by strand, followed by the multiplexing of reads. I also used a function from the ISOSEQ program to remove any concatemer artifacts. Then all the reads are aligned to a reference sequence with Blazor. Then we used our own tool for allele calling. Also for variable site identification, I double-checked our results with the ones generated with one of the PacBio programs called Juliet, and the results are pretty much the same. So using this method, we identified 62 alleles in total, 38 of which are new alleles that haven't been found before. For the first time, we were able to confidently infer MHC haplotypes for the devil, and also to do a thorough characterization of polymorphic sites within the peptide binding group of MHC proteins. Based on biochemical properties of amino acids at those variable sites, we were able to group all the alleles into 13 supertypes. So each supertype potentially have a different antigen binding preference. So all these results are quite useful for understanding immune responses against all the different pathogens, DFTD, as well as DFTD vaccines. So in summary, the long-read-based MHC typing method has many advantages over traditional methods, and obviously the protocol that we're using can be easily adapted for using on other species as well as other polymorphic gene families. So I'd like to acknowledge my co-authors, Save the Tasmanian Devil program, all the founding buddies, and my lab mates. And thank you for listening.
Thank you very much, Xuan. That's a very interesting talk. We have a question from Chris Loma. Are these mosaic CCS reads the result of PCR recombination, or do you think it's some other process?
It's a result of CCS calling using all the subreads from the same SMARTBell templates, because the forward strand and reverse strand in the SMARTBell are different alleles, actually. So they have all these different SNPs in them. And if you do CCS calling using all the subreads together, it can sometimes create these mosaic CCS read result.
Okay, Anonymous writes, I really like the name Bellerophon and a nice strategy. Have you done some similar analysis using Illumina data, and how different are the results?
We had some whole genome resequencing data for a range of devils, and I looked into MHC reads in those data sets. But the problem is you can't confidently map the reads to the genes that they came from, and the way MHC genes, so in MHC genes, basically they have these hypervariable regions and in the exons, and in the case of devil, those are the most polymorphic regions, whereas in the introns, there are very few SNPs. So you basically can't like confidently assemble the Illumina short reads into full-length alleles, because of all the introns are pretty much the same. So you can't, correctly face the alleles.
Right, thank you very much. And we'll move on to the last talk.
Great. So thank you to the organizers for the invitation to speak today. I'll be talking about a project that's very much a work in progress and not yet a completed story. So I'd love to hear feedback from any of those out there who might have been trying similar approaches. The main thesis that I'd like to argue today is that perhaps we should all consider using nucleus extraction much more routinely in biodiversity genomics because it can give us all the data types that we need to build near perfect genomes from single input specimens, large and small. So in today's kitified world, the status quo is that if we want to build a perfect assembly for, say, this rosemary beetle, we might easily need several individuals, one for agarose plug extraction and optical mapping, one for high molecular weight DNA extraction and long-read sequencing, one for RNA extraction with Trizol, and one for high-C library prep with a kit from a manufacturer like Dovetail, Arima, or QIAGEN. This might be fine, although I'd also note that, in my opinion, the impact of using multiple specimens for different data modalities, especially in heterozygous species, hasn't really been rigorously studied. Also, if you're lucky and your specimen is large enough, you could probably dissect it and get 2 or maybe even 3 birds with one stone with these commercial methods. However, with much of biodiversity, the requirement to have many specimens is limiting. In most communities, the rank abundance curve is highly skewed, such that many species are quite rare. Also, with very small specimens like this myofaunal flatworm here, which is about 1/4 millimeter long as an adult, dissection is simply not feasible. So in thinking about this problem, I found some inspiration from the flow cytometric genome size estimation literature. In this field, people use different buffers and extraction methods to convert specimens into a pure suspension of intact nuclei that have been stripped away of any cytoplasm. These nuclei then can be stained and run alongside a standard of known genome size. And as long as the staining is proportional to DNA content, the genome size can be estimated as a fluorescence ratio. I want to know if it's possible to adapt these methods on kind of a microcentrifuge scale for other purposes as well. So the broad idea is take your specimen, homogenize it up front if it's very large, and convert it into a nucleus suspension. Then centrifuge, and the dense nuclei will form a pellet, whereas all the cytoplasmic contents will remain in suspension, in solution. In theory, the supernatant of the first wash of this process should contain intact RNA, which we should be able to extract. We might then also purify the nucleus pellet with a few more washes or other steps to further fractionate the nuclei from any other dense co-pelleting bodies. Incidentally, I'll also point out this is a really good way to extract high molecular weight DNA from difficult samples like many plants and mollusks that have high amounts of polysaccharides and mucus, because it allows you to physically separate the nuclei from the mucus, which remains in solution. Finally, once we have a pure nucleus suspension, we can split this liquid using 1 fraction for genome size estimation, 1 for DNA extraction, 1 for high-C, and maybe 1 for a more sophisticated agarose extraction. So a key step in getting to this pure nucleus suspension is disrupting the specimen if it's large. There are many ways to do this, from downsing to cryodisruption to a motorized pellet pestle to even just simple trituration with a pellet or a vortexer. I'm still kind of optimizing this part of the protocol. A general lesson that I think I've learned is that to get good pure nucleus yields, you need to be as gentle as possible, letting the buffer perfuse fully and minimizing shear forces and especially grinding. With soft-bodied small organisms, simple pipetting or vortexing is often enough. With insects and probably most animals, gentle downsing is also a really good strategy. In Tree of Life, where throughput needs to be very high, we also use a cryoprep, which seems to work pretty well. And I've also noticed that with plants, some kind of cryodisruption does seem to be necessary. And I think this is going to be a general rule for any organisms with thick cell walls. There are lots of nucleus buffers out there, and I can't really get into the details of them here. I just note that the components that they share generally include non-ionic detergents to disrupt the cell membrane but leave the nucleus envelope intact, organic buffers to hold the pH at modest alkalinity, chelators and reducing agents to inhibit DNases and RNases respectively. Salts and sucrose are generally used to regulate ionic strength and osmolarity. And then a really common component are polyamines, these kind of elongate charged molecules which bind the DNA backbone and precipitate that DNA or physically condense chromatin. And this is, in my opinion, a really crucial thing. You'll also sometimes see components like PVP or protease inhibitor, which I see as kind of optional for depending on the tissue, as long as you're working on ice. I also want to emphasize that the process of going from a liquid homogenate to a pure nucleus pellet does remain a bit of an art still. I commonly use a series of filters to physically remove debris progressively, and I like these PluriSelect mini strainers, which fit on both 15 mil Falcon tubes and normal Epi tubes. However, even with stringent filtration, some further purification is usually necessary. With many animals, simply pelleting the nuclei and gently resuspending that pellet in fresh buffer several times is enough to accomplish this. You'll often see in the first wash, the supernatant is kind of colored or opaque, and there may even be a lens containing other organelles like mitochondria. And over many washes with gentle resuspension, those disappear, leaving you with a translucent nucleus pellet with nice, neat edges. For some tissues, though, that's not enough. And that's particularly the case with plants, where I often see trichomes or fragments of them co-precipitating, or this, I had a fecal problem with the snail. In those cases, probably density gradient centrifugation is a very useful thing, although it's laborious and a bit lossy. Of course, it's much easier to extract DNA from a pure nucleus pellet than it is from an entire organism. I've generally been using a salting out method, but probably many other methods would also work for this. One thing I've found quite helpful is to pre-wash the nuclei before extraction in Galbraith's buffer, which has a high magnesium concentration, but no polyamines. The magnesium displaces the polyamines, which is good because even small concentrations of these can make it really difficult to get high molecular weight DNA back into solution. In general, I found that even with really small pellets, say the size of a lentil, you can get many hundreds of nanograms of DNA. Larger pellets easily give you multiple micrograms, and this DNA is, of course, very high molecular weights, protected from shear forces during most of the extraction. However, even with super tiny pellets, like you get in many myofauna, it's possible to extract with high efficiency so long as you use a carrier. I like yeast tRNA. I've also managed to demonstrate, at least to my own satisfaction, that we can get good cDNA libraries from the supernatant, as I've mentioned. I use oligo-TT magnetic beads to do this, and the only trick really is that before adding the beads, you've got to dilute the supernatant in a lysis buffer that has a lot of salt and SDS and proteinase K to displace polyamines and disrupt any protein-RNA interactions that could prevent hybridization. I've gotten good yields from this method using both large samples that don't require any amplification, like the rosemary beetle, and from tiny myofauna that require PCR. And to my eyes, at least, the cDNA distributions look quite nice, with mean sizes of 1.5 to 3 kilobases and a tail of larger transcripts. I've also done one pilot experiment, which I think shows at least the principle that these nuclei preserve chromatin structure and nucleoprotein interactions enough to let them be compatible with Hi-C. So to demonstrate this, I used a flatworm, which I can find in relative numbers from the can nearby. It's this Rhabdocele castrella truncata, that's 0.5 millimeter there. And I've already made a contiguous assembly from a single individual using a long-range amplification method, which I'll be publishing soon, I hope. And to further test this, I took a new individual, made a nucleus suspension, and took half of those nuclei and fixed them with formaldehyde and processed these using a slight modification of this published low C method for very low input high C. A few weeks ago, I got back some MiSeq data from this run. It shows, admittedly, a relatively high PCR duplicate rate of about 50%, and the coverage isn't great to begin with, about 17x. However, the read pairs mapped to the genome clearly show evidence of very long-range interaction out to tens of megabases, as we expect from a good Hi-C library. I've also tried to see whether these reads have value in scaffolding, and the answer seems to be yes. I've used SALSA2, and with that, I was able to bring my primary kind of preliminary assembly from, which is 230 megabases, from an N50 size of 3.1 megabases to a scaffold N50 of almost 44 megabases. So that's really what I wanted to say. You should leave this with the impression that there's definitely still a lot of work to do. In particular, it remains to be seen how easy it is to use these nuclei for something like optical mapping. I think there's also a lot of major unexplored potential for the use of flow cytometry as a tool in this world, both for genome size estimation and, for instance, for separating mixed samples like symbiotic organisms. Disruption methods are still also needing some optimization, particularly for many plants and perhaps like Dicyozoa and myofauna. And of course, scaling is going to be a major question here. So far, I've been approaching this a bit like a hipster barista pulling espresso at different pressures, but at Tree of Life and similar initiatives, we need to be Starbucks, although hopefully making a better cuppa. So thank you for listening, and keen to have any questions.
Thank you, Chris. That was great. I mean, this work is very, very important for the Tree of Life projects as they scale up. So one question is, do you have a version of the protocol online yet? Is that to come?
Not yet, hopefully soon. I would like to publish this as a tiny little manuscript, maybe one for smaller inputs and one for larger.
Mm-hmm. And if a sample is snap frozen, would this still be compatible with the protocol?
Yeah, I've actually been using either completely fresh, still living specimens, sorry all the invertebrates I've killed, or flash frozen. So that's an ideal place to start. It does remain to be seen if things like RNA later or ethanol preserved specimens are fully compatible with these methods.
And the method will lose the mitochondrial DNA. Can we still recover it from the supernatant or of the nucleus pellet?
In theory, yeah, I've not tried that. I have sequenced some RNA from that, and you see lots and lots of mitochondrial transcripts confirming that there's that separation. It might be a good thing though, right? In many organisms, you have really high read coverage from plastids and mitochondria, and those are reads that you don't use for assembling the harder nuclear genome.
Cool, thanks, Chris. And thank you everyone for a good session. And I think we need to leave it there so people can get ready for the next one.